Today I'm going to present some basic knowledge about the photoreceptor and the phototransduction. First, let's take a look at the whole structure of the eye. First, let's take a look at the whole structure of the eye. The light comes through the cornea, then passes through the anterior chamber, then focused by the lens, then goes through the vitreous chamber, then hit the retina. The retina is a seat at the last back of the eye. When the light hit the retina converts the light signal into a electricity activity, then convert with then sent to the brain through the optical fiber. So actually, retina is like a piece of a CCD. For the primate eye, there is a very specialized structure called fovea. At the fovea, there's no ganglion cell or bipolar cell. There's only photoreceptor layer and a very special photoreceptors type. That's the cone photoreceptor. Cone photoreceptor function at the daylight, and although it can only be activated at quite stronger light, but this is what like when for our human being that's used dramatically. So here let's take a look at the anatomy of the retina. Here this is the inside of the retina, and here is the outside, and close to the choroid or sclera. Light comes through the whole retina, then hit the photoreceptors. There's two types of photoreceptors. One is a cone photoreceptor, another one is a rod photoreceptor. Rod photoreceptor function at dim light, and the cone photoreceptor function at bright light. For human, or like other mammal, for example, mice, there's three types of cone, and this three type is determined by functional. Those cones are like a short wave or blue cone, and the middle wave, the green one, and the long wave, the red cone. This is determined by the types of the protein that sends the light uh, particle, the photon. After capture the photon, the cell will send electricity activation to the bipolar cells, and this step is being modulated by horizontal cells, and then bipolar cell send signal to ganglion cells, and the cell synapse connection is at the inner plexiform there, and these connections are modulated by microglia cells. There's other cell types provide nutrition or supportive on other like immunosurveillance. These cells are glial cells, Muller cell, microglia. And the ganglion cells will send the electricity signals through the optic nerve to the brain. So overall, the light comes through in this direction, and the light signal that is transmitted from this direction. Photoreceptor is a receptor of a photon. It's a cell type. The light hyperrides in the vertebrate photoreceptors is called dark current. The reason that it's called dark current is because it sends signals or is activated when the light turns darker. And at this step, there's no action potential is involved. Cycling GNP gated cation channels are responsible for the initial change for the electrical activities. There's a consistent relationship between light intensity change and the rate of transmit release from the photoreceptor terminal. So the photoreceptor response is a response to the light intensity change. When the light intensity goes dimmer, goes smaller, then it will release more neurotransmitters. Here, this picture is show that the photo spectrum that for like a human being, we can see from around um 350 nanometer to like a 700 nanometer. And between this, within this range, the peak photon spectrum response of the rod is at 496. The cone is the blue cone is 419. Green cone is 531, and the long wavelength, the red cone, is actually looks like it's a yellow cone, and the peak response is at 559 nanometer. And for the rods, and already functional at a much dimmer light than a cone photoreceptor, and the peak spectrum is 496, very close to 500 nanometer. In previously, I said that the retina is really like the CCD of a camera. The light signal hits this piece of film, either the CCD from the camera or the retina, then transmit to from light energy to electrical signals. So actually, for because the photoreceptor actually responds in at a relevant light intensity change. So the same light flash may produce different flash response depending on the background light or or the previous condition within the photoreceptor which is within. In this lecture, now we are going to move on to the phototransduction. Phototransduction is a process by which light is converted into an electrical signal in the eye. It's a biological conversion of a photon into an electrical signal in the retina. It occurs primarily in the photoreceptor. There are many diseases that related to the phototransduction dysfunction, and there are over 300 genes linked to retinal disease. And about half of these genes are selectively expressed in photoreceptor. So, do you know any condition disease affecting phototransduction? I can raise some examples. For example, vitamin A deficiency, since vitamin A is ligand for the opsin. So if there's a vitamin A deficient or the body or bloodstream cannot provide enough vitamin A, then it will cause blindness. Um, and other conditions, for example, like color blindness, there's some dysfunction or mutation of the cone opsins, and that will cause color blindness. But since the rod photoreceptors still function, or other type of cone photoreceptors still function, so still we can see something. But the patient cannot maybe may not discriminate, for example, like red and green. Phototransduction is follow a very popular sensory transduction pathways. That's the G protein. G-Pro signal pathway because it's a cascade, so it can amplify this signal dramatically. So generally said, the energy photon being captured by G protein coupled receptor, and here is like a different opsin or rhodopsin. The rhodopsin or being there's a conformation change of a rhodopsin that activate transducing, I'm sorry. Then transducing will activate the PDE, that's a phosphodiesterase or hydrate a cyclin GMP. And this cyclin GMP uh, decrease the concentration will close the channel of the CNG channel. CNG channel is a cyclic nucleotide gated channels, and this will cause the reduce of fluxing of the this will cause the channel cause and it will cause hyperpolarization. With the hyperpolarization, then the synapse release of the neurotransmitters, its glutamate, will be reduced. Here shows a diagram for the photoreceptors rod and cone. Rods and cone both have several functional specialized sub area like outer segment. The cone's outer segment is look like a cone, and rod photoreceptor is longer and has more discs. An outer segment is embedded into the pigment epithelium, and the ligand that is the retinol will be recycled between the pigment epithelium and the photoreceptor. Then the inner part, like in the inner segment, and it is where the cell nucleus located and a lot of mitochondria that provide energy that is needed for the phototransduction. And the synapse terminal will form the snapped connection to bipolar cells, then send the signal to the downstream the bipolar cells. And the photoreceptor and the G protein coupled receptor rhodopsin is located at the discs in outer segment. The discs is floating on the discs, and here it shows that a rhodopsin molecule. A rhodopsin molecules that combine the two parts. One is a protein, it's called opsin, and the ligand is binding within this 7' segment. And here it shows a 11-cis retinol, and this whole um, complex is called rhodopsin. Here is a picture of a rod and cone, and we can see that the name is coming from that shape. For rod, it has a longer outer segment. It can capture more light because when the light goes through the whole cone, so whole rod, there's a more chance being captured by the rhodopsin floating on the discs because it has a longer segment. So that's why it's more sensitive and functional and during the night. Since there's only one type of rod, it can only detect black and white or grayscale. And the cone photoreceptor is less sensitive as a much shorter outer segment. So we can see here, outer segment is only this part. This is already the inner segment. That's the mitochondria located. But it's a fast response. It can detect from a color because there are three types of different cone. So the difference of the signal between the different cones, and we can tell what's the color it is. It functions during the day. So on the right side here, it shows that um, in the future, when we discuss about the phototransduction process, and we'll use this image to talk about it. And this structure and is represent the discs. And this membrane is the outer membrane of the photoreceptor, and the outside is the extracellular matrix in the outside. And this disc is floating in the inside. But in the system of the disc, the composition is quite closer to the outside. And here is the cytosol. The visual pigment and the rhodopsin molecule is a light absorbing ones. And it's on the outer segment membrane discs. And there are two components. One is the protein opsin, another one is organic molecule 11-cis retinol. 11-cis retinol is coming from vitamin A. There's a different opsin and different photoreceptor. Rhodopsin in rods, and three types of opsin for color vision in cones. So here we can see that the rhodopsin molecules, its C terminal is located within the discs, and the N terminal interact with the downstream signal pathway, that's the G protein, also called transducin. It's within the cytosol. Here is the organic molecule, the 11-cis retinol. It's coming from the vitamin A after two steps of chemical change. The first step is isomerase or transretinol change to cis-retinol. So the difference is here. And the second step is the oxidation, and this part is changed to this part. And after captured by a photon, the 11-cis retinol will undergo automatically conformation to change to all transretinol, and all transretinol will dissociate from the opsin, then recycled by the RPE. Rhodopsin molecules is a 40K Dalton protein with 348 amino acids. It has 7 transmembrane domains and many glycosylation sites, and is included as a G protein coupled receptor. from the top of the view, or we can see when we take a look at from, let me see which part. I think probably it's either way. That's just a change at 90 degrees. Then we see that the 11 cis retinol is sit in the center of this protein. And the binding site of the 11 cis retinol is at the site of lysis 296. After capturing light, the conformation change of the 11 cis retinol will induce a rhodopsin's conformation change. And this rhodopsin conformation change will make it activate the G proteins. And here's a question. How many visual pigment in the retina? And we will know that there are about 40,000 visual pigment on one disc side, and its diffusion generally set like relatively freely around the discs and at the speed of 1.5 microgram square per second. And in total, there's a 16.8 quadrant visual pigment in rods. So for such a dramatically number of the visual pigment, it's really hard for the photon to escape from all these disks. After the light-induced retina changed from 11 cis retina to ultrasense retina, rhodopsin is being activated. Here we use this bump to show that a conformation change. So the details on change of rhodopsin is that the capture light and you will undergo a lot of conformation change. And each different conformation will be the most sensitive light wavelengths will be changed for each conformation change. So after capture the photon, the rhodopsin will change to photorhodopsin and several steps. Then another more stable status is the metal rhodopsin 2. And then go back to a dysfunction of the opsin, and all transretinal will be dissociated from the opsin, then transport into the RPE cell, the retinal pigment epithelium cells, then change back to 11 cis retina, then transport out of the RPE, then entered into the photoreceptor, then binding with the opsin to form a functional rhodopsin that's ready to capture a photo again. The visual pigment is capture light is the first step of phototransduction, absorbing photon and convert the energy to rhodopsin opsin conformation change. One visual pigment molecule absorbs a photon at a time. A single photon can excite a rod enough to make it emit a significant signal for humans to see. Yes, there is a question about how many photons need to being captured by one rod photoreceptor to provide to stimulate a photoreceptor activation. Based on the experiment, it shows that a single photon can work. There's a dramatically difference between a single photon or two photons. For the possibility of that one photon um, arrived at one rod photoreceptor is much higher than the two photon at the same times arrived at the photoreceptor. So this we can know that the rod photoreceptor are really sensitive due to this G-protein signal pathway. The zillions of visual pigment packed into outer segment of the photoreceptor. So it's almost like it takes no time for a photon to hit the visual pigment to activate it. Then what's the next step? How this capture of a photon to convert into electrical signal. Visual pigment are located on membrane disk in outer segment. Light-induced current change is recorded on the plasma membrane. The time course of a single photon response is on the order of seconds. Although it takes no time for the photoreceptor, or we can say for rhodopsin, to capture a photon, but this signal will take like seconds to start to transmit to a downstream uh, to the downstream bipolar cells. So we can say that within a dim light or very dim light, the eye's response is not so fast, and it takes more time for a rod photoreceptor the signal transmitted to the brain, and cone photoreceptor is more fast. So the time course of a light signal, we can say, is like seconds. The calcium and cyclin GMP is play a very important role in the phototransduction. In the history of vision science, people are searching for an internal messenger. So people were thinking that which second messenger for visual activity. There's a very long time, all the people think that the calcium is function as the key messenger. And there are a lot of experiments to demonstrate that without the calcium, it's not functional. But when we're talking about the messenger, is to convert this take this signal and to directly to open a channel to change the membrane potential of the photoreceptor. Unfortunately, it's not the calcium. Although calcium plays a very important role, but this key messenger is not calcium, it's cyclin GMP. And later we'll know that because the channel is cyclin GMP gated channel, it's not a calcium gated channel. Since it's a cyclin GMP gated channel, so the key messenger is cyclin GMP. So in the history of the science, there's a two key research finding. The first one is a reduce external calcium concentration, increase light regulate current. And the second one is that the downstream of the G protein, the transducin, is the second GMP phosphodiesterase, the PDE6. So the light will cause a G protein, and the downstream is that the G protein will activate the PDE, and the PDE will hydrolyze the cyclin GMP and reduce the intracellular internal cyclin GMP level. CNG channel is a tetramer, and so it's composed by 4 subunits, and each one required to be bind a cyclin GMP to cause this the whole channel open. So the cyclin GMP getting conductance suggests that at least it required 3 to 4 cyclin GMP binding. Then the channel can be activated. So after 11 cis retinol capture a photon and change to all-trans retinol and cause a conformation change of rhodopsin, then this conformation change will bind to a G protein, the transducin. There's a super big family of this G protein, and for the transducin, the alpha subunit is alpha I. That means that it will cause the decrease concentration of a cyclin nucleotide, the decrease of cyclin MP or cyclin GMP. And the photoreceptor is a cyclin GMP. G protein is guanine nucleotide binding protein. So at the resting state, it always binds GDP. Involving transmit signal from variety stimulate the outside cell to the internal. It often coupled with a 7 transmembrane domain. That's the G protein coupled receptor. In this case, it's a rhodopsin. It has 3 kinds of subunit: alpha subunit, beta subunit, and gamma subunit. And the beta and gamma subunit are more close, and they always bind together. And after activate, the alpha subunit will be dissociated from the beta and the gamma subunit. The inactivated state, these 3 subunits are binding together and are binding GDP. And after activate by all-trans retina, activated the rhodopsin, and it will dissociate with the GDP and binding with the GTP. After binding with the GTP is an activate state, and it will dissociate into 2 parts. And an alpha subunit of G protein, we're going to activate the downstream effector, which is the PDE6. So we're just talking about transducing, and that's a G protein. And the next step is the PDE6. Within the 200 milliseconds, the activated rhodopsin. So in the foreign structures, I'm going to I'm going to use a rhodopsin star to indicate it's an active state of the rhodopsin. Rhodopsin star can activate about 150 transducin in over a duration time of the excitation phase. PD6 is being activated by uh, the alpha subunit binding with GTP. PDE6 is actually is looks like um, it has an alpha subunit and of almost the same size a beta subunit, and this subunit is being locked by the two gamma subunit. And for each like alpha or beta subunit, there are two parts. One part is the GAF domain, and another part is the catalyzed domain. For the GF domain, it will bind the cyclin GMP, and the cat domain it will hydrolyze cyclin GMP. So as a function two part, one is binding, another one is like doing the work of hydrolyzation. Phosphodialysis or PDE is a superfamily. It has 12 isoforms or subtypes of of enzyme that can break a phosphodiester bonds. A subgroup of PDE that degrade cyclin G AMP and or cyclin GMP. The key regulator of signal transducing mediated by this second messenger molecule. PDE inhibitor are used in clinic to treat various diseases. For PD alpha and beta subunit, as we just discussed about that, it has the GF domain and CAT catalytic domain. Here is binding by the GF, then enzyme hydrolyzed at the CAT domain. The gamma subunit is a regulatory subunit. So after So generally functional is said that this PD6 will hydrolyze the cyclin GMP to GMP. And if totally removed of the gamma subunit, and it will continuously being activated and do the job of hydrolyze cyclin GMP. In a natural condition, the gamma subunit is being kind of like being binding to the alpha, binding by alpha subunit of the G protein, and it's kind of like release the alpha and beta subunit of the PD6. Sometimes it's the alpha subunit is being released, or sometimes it's beta. But if there's a lot of activated transducing, then both alpha and beta subunit can be released and do the job of hydrolyzed cyclin GMP. Phototransduction. So here the signal cascade is come from rhodopsin and capture light, then activate the rhodopsin star and activate the transducing, and transducing is separate and alpha subunits of transducing to activate the PDE6, and PDE6 start to hydrolyze the cyclin GMP to the GMP, and the concentration of the cyclin GMP will be reduced. It is a simple signal relay, and I think actually it's not. So here is like kind of like uh, recycling and closed loop of this signal pathway. Rhodopsin capture a photon and change it to rhodopsin star. Then binding with the transducing. Transducing will dissociate with the GDP and binding with GTP to be the activated state of the transducing GTP, then activate PDE6, then PDE6 on gamma subunit, binding to the transducing GTP, then it's being activated, and then it's to hydrolyze the cyclin GMP to GMP. Then it will be inactivated, then dissociate with the transducing, and the PD6 becomes like being silenced. And here this rhodopsin is can be rhodopsin star. It can be recycled because and it can activate one PD6, then activate a second PD6 during its activation phase. So this step um, produced a significant amplification. There's two steps. The first step is signal photolysis rhodopsin generate about 150 transducing GTP. Sorry. A single activated phosphodiester hydrolyzed about 1,000 cyclin GMP. So one rhodopsin star can activate about 100 transducing and transduce 1 to activate hundreds of PD6 and will cost about 10,000 second GMP hydrolysis. So now this signal amplification from 1 to 10,000. In next step, we're going to talking about the cyclin G channel. Cyclin nucleotide gated ion channel and is a tetramer. It have 4 subunits, 1, 2, 3, 4. Like in rod photoreceptor, it's a A1B1. And for the cone photoreceptor, the channel is composed by the A3B3. A secondary channel family function in response to binding of cyclin nucleotide, cyclin AMP or cyclin GMP. It's located in the plasma membrane and it's inside the cell. It's like a channel that link to the inside of the cell and outside of the cell. Its significant in sensor transduction. It forms a tetramer of alpha and beta subunit around a central pole. Each subunit have a core region of 6 transmembrane segments. In a closed state, the calcium and the sodium ions, cations, cations cannot enter into the cell. But after binding with the cyclin GMP, more cyclin GMP will cause channels to open. So this channel is always like in the closed state and or an open state. If the cyclin GMP molecules concentration is very low, then this channel will prefer in a closed state. But if the cyclin GMP molecule concentration increase after binding with the cyclin GMP, it will more prefer to stay in an open state. So here in this image, we can consider that the cyclin GMP is like in the light within a house. In a darker phase or during the night, we turn on the light and the whole room is with like a yellow light from the light bulb. And the same as like in the photoreceptor. In a darker state, the cyclin GMP concentration is really high and the channel is open and there's a current forms flowing circuitry within within the cell across the membrane, and there's a current flow. It's also like the electricity that we turn on the light and it forms and close the circuitry, and the the light bulb will be turned on. Uh, During the day, or in the bright condition, the light is turned off. There's no yellow light from light bulb, and then the switch is off, and there's no electricity running in the house. It's the same as in the photoreceptor. During the day, the cyclone GMP concentration is low, and the channel is closed, and there's no a closed circuitry of the electricity across the photoreceptor membrane. And this is all induced transmit by the cyclin G channels. In at night or in a dark condition, there's a high cyclin GMP within the cell. The channels are open and there's a current influx. In bright condition, PD6 is being activated. A cyclin GMP concentration is being reduced by hydrolyzed, and the channel closed, and there's no current across the membrane through the cyclin GMP channel. An open of the cyclin GMP channel will cause the membrane potential depolarize. But the light will close the cyclin GMP. So when the photon is being captured by the rhodopsin on the disks, it will cause the channels close, and the membrane potential will go to more negative because there's no influx of the cations, sodium or calcium. Second GMP gated channel remains open in the dark. The calcium influx depolarize the photoreceptor, and it's called a dark current. It's like during night. There's electricity running on to turn on the light bulb. The dose-responsive curve is linear in the relationship between conductance induced by cyclin GMP and the concentration of the cyclin GMP. Free cyclin GMP concentration in the dark is 1 to 5 micromolar. So it's very high, and the resting potential of the photoreceptor is at 0. But comparing to other cell types, the most popular membrane potential for other neurons are about like minus 50 millivolt to minus 70 millivolt. So average is like minus 60 millivolt. And the current and voltage relationship in rods is the light suppressed current varies with the membrane potential. At the normal operation range is minus 35 to minus 60 millivolt, and at this condition, it's quite flat. The curve is quite flat, and this means that a little tiny change of the influx current may cause a dramatically change of the membrane potential. And we can see here, I think there's a 1 peak ampere change, and it will cause the photoreceptors membrane potential change from minus 60 to minus 20. And this is super dramatically that comparing to the change of the current. So this causes that the photoreceptor is very sensitive It's all caused by the cycling second CNG channel, the cycling nucleotide gated channel. The phototransduction activation, then we summarize is a photon change rhodopsin from to the rhodopsin star and activate the G protein transducing and dissociate alpha subunit or activate the PDE6, and it will cause the cyclin GMP concentration down, then close the cyclin GMP gated channel, and then stops the influx currents of the photoreceptor, and this was caused a hyperpolarization of the photoreceptor. The light signal is transferred from disc membrane, where is the rhodopsin located, to the plasma membrane, an outer segment of the photoreceptor. And another channel is very important in the phototransduction is the voltage-gated calcium channel. We remember that cyclin CNG channel is a ligand activate channel, and the majority cation flux in is the sodium. And at the terminal of the photoreceptor is a voltage-gated calcium channel. So its open or close is determined by the membrane potential. Do we remember that there's like very tiny change of the current can dramatically change the membrane potential of the photoreceptor? And here is the fact that as a little tiny current influx or the cation influx will cause the membrane potential, the whole cell's membrane potential change, and it will be sensed by this voltage-gated calcium channel. And at the terminal of the photoreceptor, this channel is a high voltage activated channel, CAV 1.1. It's a big family with different type and composition and complex composition of different subunits. In the membrane excitable cells and also photoreceptor, it's permeable to calcium at depolarized membrane potential. So remember, the CNG channel, it can influx both calcium and sodium, but this voltage-gated calcium channel, only calcium can influx into this channel. The voltage-gated calcium channel VGCC in the photoreceptor is a primary L-type voltage activated calcium channel. L-type means that it's open, it's not a transient, it's quite stable. So when it's activated open, and it can like stay open in longer time. But it need to be activated and at a higher membrane potential, more depolarized. Let me see. Membrane hypertension, high voltage, low voltage guide. Yeah, it's activated by higher, more depolarized membrane potential. As light-induced membrane hybridization lead to these channels close, and this close. So after captured by the photon, then so all the phototransduction cascade is that the light will cause the membrane hyperization, and this channel will be closed, and the calcium influx will be decreased, and calcium concentrations will be decreased in within the cell, and the glutamate is also being reduced. So the light will reduce the activity of the photoreceptor. But the photoreceptor is not like used only one time. It need to recycle it, and the whole system need to be restored to a previous state. Like the dark condition, there's a lot of cycling GMP and channels are open, and there's a current across the membrane when the light will close the channel and reduce the activity of the photoreceptor. But how can the photoreceptor automatically recovered, go back to the dark state? So this step is called the system restoration. Everything needs to be regulated, quenched, inhibited, or removed to restore to the normal state. All these have to be fast enough to provide the subsequent rate of photoreceptor recovery. So the time scale for the photoreceptor, like activation, seconds, recover, seconds. So we remember that when we go from like the dark room to the bright daylight, we take, I think we actually, I think we need to take like a minute to be adapted to the condition light change. For the single photoreceptor restoration, it takes seconds. The first step is the visual pigment recycling. For the pigment recycling, have two steps. The first one is the protein part, and the second one is the organic molecule, the ligand, the retinols recycling. First step, um, okay, then we start from here. That rhodopsin will change to rhodopsin star and dissociate with the all-trans-retinal. All-trans-retinal will be transported by RRBP to RPE, then be isomerized to 11-cis retina, then delivered back into the photoreceptor, and the rhodopsin start or binding to rhodopsin kinase and put a lot of phosphate onto the rhodopsin. Then after phosphorylation, the rhodopsin star, phosphorylated rhodopsin star, will be binding to the arrestin, and is at this step the rhodopsin star is totally being inactivated. And then, but how to restore the cyclin GMP concentration within the cell that required the GC enzyme. GC is guanylate nucleotide cyclase, and it can convert the GTP to cyclin GMP. But the active state is required a magnesium binding the GCAP, binding to this guanylate cyclase. If it's a calcium binding with the GCAP, then it's not activate. So just as we said that the all-trans-retinal restoration is being transported by the IRBP, then with several exchange that, like some enzyme as LRAT, then changed to an FA, then with another enzyme RPE65, then changed to 11-cis retinol, and another step to 11-cis retina, then transported go back to the photoreceptor, and this is ultra's retina uh, recycling steps. If as a functional there's a mutation dysfunction of LAT or RPE65 that will cause blindness because it cannot provide enough 11-cis retina for the photoreceptor to function normally. And one types of retinal pigmentosa, a blindness disease, and is due to an unfunctional of the RPE65 in RPE. Elenices will increase like a pumpkin and spinach and carrots. Rhodopsin kinase will be phosphorylates of the rhodopsin and at the 3 site, and each binding of phosphorylation, sorry, each binding of the phosphate will be reduced. Um, the activate function of the rhodopsin star. The rhodopsin star is quenched by rhodopsin kinase mediated phosphorylation and binding of arrestin. Rhodopsin kinase is a G protein coupled receptor kinase, and it can add the phosphate onto the rhodopsin star, N terminal, and N terminal that's been recognized by this rhodopsin kinase. The central high conserved Cadillac domain and a carbonic terminal domain with an autophosphorylation site and a consens isopolarization site. It's activated by a rhodopsin star or only binding to rhodopsin star, not rhodopsin. So it will not cause any change or do any phosphorylation on the resting state of rhodopsin. Only the activate state, that's the rhodopsin star. In the total, there will be 9 sites, but 3 of them will be totally like the phosphorylated rhodopsin star activation is lower and much lower, but not completely inhibited. And the resting or binding to the N terminal of the rhodopsin. Arrestin is about a 48 kilodalton protein with 4 subunits and multiple aligners. It also bind only with the rhodopsin star, and there's another step that it only bind to phosphorylated rhodopsin star. So first, the kinase phosphorylate rhodopsin star, then arrestin can bind to this rhodopsin star. Arrestin is a functional as a second break complete block the rhodopsin star. So here image can show we can see that between compare with these the two breaks. So if both the kinase and restin are normal, that's the Y type, and both breaks are works, and we can see that it can the light signal can go back about the 0.5 seconds and can go back to the zero current at the resting state. If the kinase is gone, or the kinase is mutated or knocked out, then this rhodopsin cannot be inactivate. If there's a kinase but there's no arrestin, there's a slightly decrease of the rhodopsin star's function, but it cannot be totally uh, go back to zero. So both breaks is required for like a totally quench the function, the activation of the rhodopsin star. And this study is coming from the knockout animal study. Another step is the recovery. In an inactive state or in a high calcium and high calcium state, and that's the resting state, or we can say there's a high saccharin GMP, there's a high calcium, the channels are open and calcium are influx and a lot of calcium. The recovery will binding to rhodopsin kinase, binding together. And when the saccharin GMP reduced and the calcium concentration reduced, there's a low calcium, then the recovery will be dissociated from the membrane and also dissociated from the rhodopsin kinase. And the released rhodopsin kinase can start to phosphorylate the rhodopsin star. Remember, rhodopsin kinase is the first break to quench rhodopsin star's activity. Recovery inhibits rhodopsin kinase and is calcium dependent, only functional at a low calcium concentration. As a light-dependent channel cross decreases the calcium concentration level and reduce the inhibition on rhodopsin kinase, which lead to a more rapid inactivation of rhodopsin star. And we also need to remember that there's a very low calcium concentration within the cell or the neural cell types, and there's a very high comparing to the intracellular. There's a high concentration of calcium outside. So the influx calcium is really a messenger of different state change. And the GTPase accelerating protein is also involved in this step. And the GAP hydrolyzes transducing alpha subunit to transducing alpha GDP. That it can dissociate the alpha subunit from GTP to GDP. And after the binding is the GDP, then the alpha subunit will again binding back to the beta and the gamma subunit, and it will dissociate from the PDE6, and the gamma subunit of the PDE6 will come back to binding to the alpha and the beta subunit, and then return back to a unstable state as in an inactivated state. And the GTPase activation that it removed the GTP from the G protein, and it also have a moderator, and that's the RGS9. RGS9's name is coming from a regulator of G protein signal 9, and can deactivate the G protein because it removed the GTP. And PDE gamma can accelerate transducing alpha interact with the RGS9. So that means that alpha subunit binding to PDE alpha and beta, PDE gamma, and this binding conformation will attract this RGS. And another part of this, another part to hydrolyze the GTP, that's the G gamma 5. That's beta 5, sorry. G protein beta subunit 5, it can accelerate GTPase activity. So both RGS9 and the beta subunit can accelerate the GTPase function within the G protein. So G protein it plays several role. It can bind to G protein coupled receptor, and it can activate the downstream like a PDE6, and in itself it also have a GTPase enzyme activity. And this activity can be like accelerate or promote by PDE gamma, by RGS9, and the beta subunit. So here is another image that we can to summarize this phototransducing. So during the dark, there's a very high cyclin GMP, and the channels are open, and the calcium and sodium are in flux continuously into the cell. So there's a high cyclin GMP, high calcium concentration increase, a high concentration of calcium binding to the GCAP can block the guanylate cyclase function, the GC's function, so you will not produce the cyclin GMP. And the light will activate the rhodopsin, change to rhodopsin star, and activate the PDE6, then hydrolyze the cyclin GMP. Cyclin GMP concentration reduced and the channel are closed, and the calcium is also reduced because the channel closed, the calcium concentration reduced, and the camaldolin dissociate from cyclin GMP channel. And after the light on the cells is a low calcium, low cyclin GMP, and the calcium concentration is decreased. And this decrease of concentration make the GCAP binding to magnesium rather than rather than calcium, and the GCAP will come into an activated state, and cyclin GS is activate, and start to produce more cyclin GMP. And after the light is turning on in light condition, the calcium concentration reduce will also cause the activation of rhodopsin kinase. Remember that dissociated with the recovery, and dissociated with the recovering and recovering, and the kinase activate, and the retinal photoreceptor star, rhodopsin star is being phosphorylated and binding together with the arrestin, then it will cause its dysfunction. So the calcium decrease as a result is to inhibit the rhodopsin star function and increase the cyclin GMP to produce more cyclin GMP. So this is kind of a negative feedback. The GC is a part of the G protein signal cascade and is activated by low calcium level and is inhibited by high calcium, and is being activated by low calcium level, and it produces the cyclin GMP, the key internal messenger cyclin GMP. So here to summarize the circles is that there's a two circle. One is a cyclin GMP circle, another one is the calcium circle. And this circle is coupled together. Cyclin GMP increase can induce the calcium increase, but the calcium's increase can automatically inhibit the production of more cyclin GMP. And the cyclin GMP binding to the channel, and the calcium is also uh, being recycled. Forms like a calcium current across the membrane that there's a continuously pump out of the calcium by the NCKX. It's like a transporter. It transport in the sodium and transport out the calcium. So the calcium's concentration is continuously like being reduced by a transporter, like to be pumped out. And the cyclin GMP's concentration is determined by the two protein. One is the synthesized one, the GC. Another one is the destroyer, the PDE6, the hydrolysis part, and the light will cause the cyclin GMP decreased by the PDE6. And this decrease will cause the close the channel, and the low calcium will start to activate the GC and produce the more cyclin GMP. So cyclin GMP's recycling is it's reduced by PD6 and produced by GC, and the internal calcium concentration is increased by CNG channel and decreased by the transporter NCKX1. And as we remember that the majority cation flux into the cells is the calcium and the sodium. And ATPase can pump out the calcium, but still more sodium in. And hey, there's no sodium always in, but we all know that the sodium is actually let's okay, then we need to check that although the calcium is being pumping out by some transporter, now how about the sodium? I think in other location maybe there's other pump or transporter that can bring the sodium out, but here we only focused on the pump out of the calcium. Okay, then to summary, this phototransduction circle is that during dark, there's a very high concentration of cyclin GMP, and the channels are open, and there's a current circuitry across the membrane, and the calcium concentration is being reduced by a transporter NCKX1. When the photoreceptor capture a photon, rhodopsin will change to rhodopsin star and activate transducin, and alpha subunits will bind into GTP, get rid of the GDP, then dissociate with beta gamma subunit, and bind into PDE6, the gamma subunit. The PDE6 gamma subunit Well, actually, I think maybe there's conformation very close between the PD6 gamma unit and alpha beta gamma subunit of the transducing, right? So that's why a little conformation change or alpha subunit will prefer to binding to gamma subunit of the PD6 to activate PD6, then to decrease the cyclin GMP concentration. Cyclin GMP concentration closed and its gated channel is closed. The calcium concentration dramatically decreased by the NCKX and the calcium concentration decreased. Then how to recover back to the dark state? The calcium decrease then will cause the magnesium binding to GCAP, and this will cause the GC to produce more cyclin GMP, and the cyclin GMP concentration is increasing. And beta subunit and RGS protein will activate the GTPase function of the alpha subunit. Then alpha subunit will dissociate from GTP, then binding to so it's hydrolyzed. So GTP hydrolyzed, then have the GTP left. Then binding after binding with the GTP, it will dissociate with the PDE6 and then binding back to the beta and gamma subunit of the transducing. And the reduce of the calcium concentration will cause the recovery dissociate with the kinase, and the kinase will phosphate 3 out of 9 sites of rhodopsin star, and then further binding with the arrestin, then the activation function of the rhodopsin is totally being quenched. So you go back to a dark state. Then the rhodopsin can binding back the 11-cis retinol that provided by RPE epithelium, then go back to a resting state of the rhodopsin to be waiting for the next step of next time's catching photon and start the photo transduction pathway and do it all over again. The system restoration there's several steps. There's ultrasound retina is re-isomerized to 11-cis retinal form in RPE. And retinal start inactivated by 2 breaks when it's phosphorylation by the rhodopsin kinase and binding to arrestin and the transducing PD in the activation by GTPase secrete protein RGA signaling 9 and cause a dissociate of the transducing alpha subunit to BD6. The restore of cyclin GMP level that's been reduced by BD6 is restored by GC. To restore the ionic balance is by ATPase and the sodium calcium exchanger. So for the activation and the inactivation, there's several circles. The circle of the rhodopsin is at the catch a photon, then be phosphorylate, catch by arrestin, then go back to resting state. The circle of transducing that it can be activated by the rhodopsin star, then dissociate from the beta gamma subunit and binding to gamma subunit of the PD6 and PD6, and then like the beta subunit and RGS9 can cause the GTP to hydrolyze the GDP, then our subunit will go back to binding again to the beta and GABA subunit. The cycle of the PDE6 is that it can activate by the transducing function to hydrolyze cyclin GMP, and with other like beta subunit of transducing, and the conformation change of the alpha subunit of transducing, and it can go back to binding to the GABA subunit of PDE6. And calcium binding GCAP will become a GCAP star, and it will activate GCs to produce a cyclin GMP. Cyclin GMP so here it shows a cyclin GMP circle that is be increased by the GC and decreased by PDE. And the calcium circle is that increased by the CNG channel opened by high concentration of cyclin GMP, and it's being reduced when the channel is closed, and there's a continuously transporter um, of the NCTX1 that can be pumped out of the cell. So that's the calcium. That's the calcium circle. So every step there's a circle between activation and inactivation. In this picture, show that the blue lines is activated by decreased calcium, and the red line is inactivated of phosphorylation, and the green line is being activated by a photon. So after we take a look at the circle, this circle, second circle, and the calcium circle, then we see that green arrow, the light will cause the rhodopsin to rhodopsin star, and transducing alpha will cause to dissociate and activate PDE6, and the PDE6 will be start to hydrolyze cyclin GMP with the light activation, and the cyclin GMP concentration will be decreased by light, and the calcium intracellular calcium concentration will be decreased by light. Then we go back to see the blue arrow. Decrease of the calcium will cause the dissociate of recovery to rhodopsin kinase, and it will cause the activation of the rhodopsin star, and decrease the calcium can increase the cyclin GMP through GCAP and the GC. And the decrease in calcium will cause an increasing of the cyclin GMP to open the channel. So finally, after seconds of the result of a decrease of calcium, it will cause the channel to open to go back for make sure that the calcium concentration is going back. So it really takes some times to recover back to the original state. And this is also the mechanism of adaptation. So this figure shows that this gray scale as shows that the light stimuli, and the color or grayscale of the line shows the background of the photoreceptors condition. For example, if a previous is a condition is very dark, so when give this light intensity stimulation, and a photoreceptor will be very activate. Okay, so the light signal and because it's spikes, so actually here we're talking about the downwards, that's the ganglion cells response, because it's spikes, right? There's no spikes in photoreceptor. There's only temperature change, potential change. But at the same light intensity, if the previous it's the background light condition is brighter, then it almost cannot give light signal. So photoreceptor, like a primate or human, or is a photoreceptor or the R type photoreceptor, its functional is really is relative. It's determined by the background. It's determined by a previously light condition. So same stimuli, same stimulus depend on the history. It can just ignored by the photosystem or can trigger at a very uh, excited response. It's all determined by the background condition or by the history. Phototransduction adaptation is to steady background light by decrease the sensitivity to light. So if the background light increase, then the sensitivity to light will be decreased. Adaptation increase the dynamic range of photoreceptor to cover about 3 order of magnitude of the light intensity. So this will make sure that at each light intensity, the photoreceptor can give a very um, high activation state once the background is different. And this adaptation is occur rapidly, involved multiple steps in phosphotransduction. It requires lower intracellular calcium. It increases GC activation to restore the level of cyclin GMP. In this lecture, we only talk about the light sensor within the photoreceptor layer, the cone photoreceptor and the rod photoreceptor. And actually, in the retina, there's RPRGC. RPRGC is a ganglion cell located here. And the scientists find that some ganglion cells have a melanopsin that can also be capture light, and it's an intrinsic photosensitive retinal ganglion cell. It's involved in circadian and other light function. It's not so fast as a lot of rod and cone photoreceptor, but it plays roles in other kinds of vision um, condition and process. And which here I'm just going to very simply talking about the photosensitive retinal ganglion cell. It's a specific type of retinal ganglion cell. It's intrinsic photosensitive. That means that it has a light sensor or light receptor that's like rhodopsin. And in IPRGC, it's called melanopsin, and cannot form any image and functional and synchronize the circadian rhythm. It can contribute to pupil size regulation and other response to ambient light condition. So it's a quite wide field. It do not like focus on the detail like line or bars. It provide a very generous light. It's kind of it also sense like the background, contribute to photopic regulation and release of melatonin from the pineal gland. some visual function in human. For the blindness patient, it can provide some visual function, like they can sense of like it's a little dark or little light, but they cannot see anything. But people think that that's the instinct, but actually that's because of the function of the RPRGC. The end. Thank you.